section 3 ecology, which is always really fun. And I'm going to introduce our first speaker, Dr. Morgan Gorris, who is going to present updated ecological niche model of Coccidioides using compiled soil samples. Dr. Gorris.
Excellent. All right, thank you so much. My name is Morgan Gorris. I am a staff scientist at Los Alamos National Laboratory and excited to share a team project on creating an updated ecological niche model of Coccidioides using compiled soil samples from all of our collaborators. So this is a collaboration between Los Alamos National Lab, Northern Arizona University, TGen, New Mexico State University, Cal State University Bakersfield, and the Washington State Department of Health. So we have a pretty good understanding that valley fever is a disease endemic to the western part of the United States, but still not all states are required to report their valley fever cases. So the map on the right-hand side shows, as of 2025, what states are reporting. In green, we have states that are hypothesized to be endemic by the CDC and do report cases. In blue are states not endemic, but still report cases. And in yellow are states that are hypothesized to be endemic, but do not report their disease cases. Now, disease surveillance is really critical for understanding what populations are at risk for contracting a disease and measuring other disease dynamics, including whether or not the disease is spreading. We also know that increased disease surveillance increases disease awareness, which leads to reduced time to diagnosis and then ultimately better health outcomes. So to exemplify the importance of reporting, if we assume that the state of Texas has a valley fever incidence rate that is 10% of that in Arizona, that means about 4,200 cases of valley fever might be going unreported in Texas each year. So we're hoping that an updated distribution map could help motivate disease reporting. So historically, creating niche models for Coccidioides have been challenging because soil sampling is time and labor intensive, especially processing the samples after you collect them. And Coccidioides has been hard to find. We know you can go out into the field, take a positive soil sample in one spot, go a meter to the right, take another sample and get a negative. So folks have moved forward with niche models for Coccidioides and using other approaches. Some have been based on the case surveillance data or epi data, but this is subject to the same biases that the case data are, so including like travel-related cases. Other folks have moved forward with soil samples from a limited geographical area, but this results in higher uncertainty when you extrapolate to areas beyond your region. So thankfully, there have been more and more efforts in recent years for soil surveillance, including some of the familiar faces we see in the photo, providing more data to set out to meet our goal to use soil samples positive for Coccidioides to inform where valley fever is endemic, ultimately to motivate disease surveillance efforts. So the tool that we use here is called MaxEnt. It's maximum entropy modeling. It's a machine learning method or a statistical method to predict the long-term species distribution. So it takes two forms of input. First is occurrence data. That's your literal latitude and longitudes of where the species is present in the environment. And second is gridded environmental data. So any type of climate factors, land use factors that you think are important for structuring where that species is in the environment. And what happens under the hood is that MaxEnt goes in and takes a sampling of your background environmental conditions to understand what your geographical area looks like. So let's say in this hypothetical example on the right-hand side, we feed it in temperature, which is on the x-axis, and specific humidity on the y-axis. The model goes in, samples across the map all of these data points, and maybe finds a positive relationship between temperature and specific humidity. So our background data here are in the gray circles. And then it compares that background data to where you have found Coccidioides, or the species. So that's called our occurrence data, and that is in the red diamonds. And so it notices that it's in areas that have lower temperature, lower specific humidity, and it ends up drawing an environmental niche, or the orange oval. And that would be the kind of mathematical region where we would expect to be suitable for Coccidioides to grow. Now we can take that information from mathematical space and then map it out into geographical space. And so our output is a map of locations suitable for Coccidioides. So here we're going to look at Coccidioides as the genus. We didn't separate by species, but we gathered positive soil samples from both our collaborators. Some of these are unpublished, and the NCBI. The positive samples span from 2004 to 2023. And then we deduplicated and thinned the data to make sure we weren't over biasing the model to match the resolution of our environmental data. So it's at a 4-kilometer grid resolution. After thinning, we ended up with 49 positive soil sample sites, and those are depicted as the red circles on the map. I'll take a moment to highlight that New Mexico's on the map. So Dr. Adriana Romero Olivares at New Mexico State University, with the support of Dr. Don Natvig at University of New Mexico, have been conducting soil sampling and have found positive outside of Las Cruces. So Don presented this last year, but I just wanted to bring it up again because we're a state too. Okay, so then we grabbed a bunch of different environmental data that we think might be important for structuring where Coccidioides is. We did do some down selecting of the environmental data. And so currently what's going into the model is 5 different climate variables, 5 different land cover types. One of them is shrubs, which you can see at the top map as an example. 12 different soil property variables. Again, pH is the example in the middle. And even a map of rodent richness. So this is going to be the number of species you would expect to be present in a given grid cell. And the rodent richness map is on the bottom. We take all of this data, we resample it to that 4-kilometer grid square resolution. We test a variety of different parameters within the model. There's a lot of different tuning metrics. We ultimately test about 42 different models. We do a best model selection process that's previously been published for other applications. And then we get our final model configuration, and we run it 10 different times to get our average output. Now the output comes out quite literally on a 0 to 1 log likelihood scale, but I like to interpret this as low to high relative habitat suitability. So this is our result map. We're looking at Coccidioides relative habitat suitability, where the darker red colors are the areas that we would expect to be most suitable for the presence of Coccidioides. So we can see our known hotspots lighting up in the San Joaquin Valley, areas across much of the state of Arizona, around the Las Vegas area of Nevada, the known endemic range up in Washington State. And then some other interesting areas pop out on this map. The Snake River Valley of Idaho is clearly highlighted. Albuquerque, New Mexico is that really dark dot that you see in the center of the state. There's patches around Texas, and this interesting kind of swath of medium habitat suitability that extends up through the center of the Great Plains, which I think is fascinating. So from this, we also get a measure of how important each one of these environmental variables were in structuring this output. If you added up all the variables, you would get this measure of 100% importance, and I just put the top 5 here. So they were soil moisture, shrub land cover, minimum temperature, precipitation, and soil organic matter. Now, since we run the model 10 times, we can also assess a spread in our output. So for each one of these 4-kilometer grid cells, we report the range in the estimated habitat suitability. And this gives us an uncertainty metric in our measurements. So although that area in the central Great Plains is really fascinating and came up as kind of low to medium suitability, it's also the area of most uncertainty on our map. So this would be a really fascinating area to target for future soil surveillance and potentially keep an eye out for in case surveillance. So ultimately, I think what we find from this is that there's evidence that more states might be endemic than currently recognized, and that all Western states should consider reporting their valley fever cases. So again, the niche models on the left-hand side, I'm bringing back up what states are currently reporting valley fever on the right-hand side. This is showing support that there is likely areas in Idaho, Oklahoma, and Texas that could potentially harbor Coccidioides. So in conclusion, based on pathogen presence, there's medium to high levels of habitat suitability in 13 different states across the Western US. We really hope that this map can help guide future soil surveillance and epi-surveillance and serve as encouragement for the 3 non-reportable states to consider reporting. So I have to take a moment to greatly thank our collaborators because this would not be possible without their surveillance efforts, including those at Northern Arizona University, TGen, NMSU, Cal State University, Bakersfield, and the Washington State Department of Health. So my contact information is at the bottom and happy to take questions if we have time. Thanks.
Great talk, Morgan. I just want to give context to those of us, particularly in California and Arizona, where we think, why don't these other states report? So we've had multiple interactions with the Texas State Department of Health Services. And it's really interesting. It's a bureaucratic inertia. It isn't political. There are a lot of counties in Texas. When we first approached them, they had the first human case of avian influenza. Now they have a measles outbreak. So it's very difficult. So it's got to be an iterative process. And it's not just saying, well, yeah, there's another disease you need to be monitoring because state health departments are very burdened now. So adding that on. And to me, Texas is the clear one because we know it's endemic. And even there, it's been a struggle and we're continuing to try to get them to make it reportable, but again, it gets into workload. And it's not that they're against it. It's just that they have so many other things to do.
Mm-hmm. Thanks. Hopefully, this can bring more money to the state to help out.
Morgan, that was great. It's really nice to see when you see things that happen and they overlay each other so nicely that it looks really good. But I'm curious, and I've thought about this for a long time, in the setting of John Taylor and Bridget's model of small mammal reservoirs, and then I see these soil samplings and soil that's good for Cocci to be in. But isn't it really about where the rodents' burrows are? More so than, like, do you weight that? Because in that list that you had, nowhere was there rodent in your predictive model at like your top 5 or 6.
Yeah.
Then that was 87% of your data on your regression analysis. So do you think maybe that needs to be weighted more by the small mammals that are present in that area, that where they live is where you're more likely to find it in the soil?
Yeah, this is a tricky question because rodents are ubiquitous, and it doesn't seem like Coccidioides has a favorite rodent that it picks on. And so if it were just purely rodents, why wouldn't Cocci be found in the East Coast? And so there's got to be some type of climate control plus the rodents.
Right.
And I think there's a lot of interplay when you start looking at the habitat types and what's what's causing the habitats to be there is ultimately the climate conditions. And then what's causing the rodents to be there is ultimately their resources, so the habitat types. And so there is a lot of interplay that happens in ecological studies. And our map that we found was species diversity. It would be really hard to find like a rodent prevalence map, but ultimately that would be a way better metric here. Yeah, how many rodents total, I think. It's a good question. Yeah, thanks.
I want to chime in too.
Chime in.
So
One more minute. So
I
quick
think
time.
richness is the wrong thing. You said rodents are everywhere. And they're functionally the same, the species. And so I think you should be saying presence of any burrowing animal. And there are a lot more of those in the Western United States than there are in the Eastern United States. Species richness didn't get in your model because they're everywhere. It's probably not
Right.
important. So it's a
Yeah.
functional thing. It's the burrow that's important.
Just to
Let's tag team, man.
additionally chime in.
Okay, come on.
I would say rodents are not functionally equivalent, right? Like, so there's
Well, some of them
there's like
are, for
there's
sure.
going to be like 45 different species of rodents in that range or more,
To
and
the pathogen, they're functionally the same.
maybe, but yeah. So like the ecology of individual rodent species would be relevant. And so I was going to ask, like, instead of doing rodent richness, you do like the presence of different rodent species and iterate through your models and see which rodents overlap with the soil concentration. That would be the way
Yeah.
to figure it out.
We started to pull out the important rodents that Dr. Salazar Ham has found. And a lot of them were widespread across the Western United States and really didn't give us anything to go off of, like, ooh, yeah, that aligns with, the Phoenix area. My time is up. So we should talk more about rodents. Thank you.
Thank you for a great presentation. There are 2 more questions online that, can we connect with the online questions afterwards so that you could answer those? And we will make sure we have a rodent session next time. I'd like to introduce our next speaker. Catherine Walter is going to present leveraging elevation gradients to characterize the distribution of Coccidioides in southern Utah, a field and modeling study.